So, good afternoon, everybody. and Today we're going to be carrying on our discussion about metabolism, about cellular metabolism. And in our last class, we learned a lot about the nature of energy in the cell and the interplay of energy and matter. And before we carry on, seeing exactly how the cell derives useful biological energy from foodstuffs, for example. Let's just remind ourselves about the kinds of cycling, the kinds of interplay that we witness when we consider energy and matter and their relationship. First of all, to remind you, the law of thermodynamics, which states that energy cannot be created, it cannot be destroyed, but it can be transformed. It can be transformed from one form to another. Biological systems require energy to be presented to them in certain useful forms. Second of all, in transforming energy from one form to another, which happens the whole time in biological systems, is one of the principal driving forces of the metabolism. Every time energy is transformed or transferred, it is done with a certain level of inefficiency. Some of it is always lost as low-grade energy, usually in the form of heat, which in biological systems cannot be used. It cannot be used to drive processes which will do work. So there's a continual loss of energy as it passes through all of the transformative and transferring reactions that constitute metabolism. Energy is continually lost. And I pointed out to you, put your hand on your arm, feel that warmth. That warmth is the result of the loss of energy from heat, as heat, from these kind of inefficient reactions taking place in your metabolism. All of that heat generated by the planet ultimately is radiated out to space and is lost. So energy passage through our biosphere is one way, in and out. And energy has to be continually be replaced in order to drive the biological systems that make up the biosphere. That's point number one. Point number two. Biological systems also require matter. They require atoms in order to build their biomolecules, and those biomolecules are what drive all the processes of metabolism. So we continually need a source of molecules, but our planet is finite. It is a closed system for molecules. So we recycle our molecules through birth, death, decay, dissolution of major macromolecules into simpler compounds, which are then recycled through the biosphere. So let's have a look and remind ourselves of exactly how this is done. First of all, fortunately for us, we have a continual source of energy, which is the sun. And the sun's energy reaches us in the form of light, which biological systems can use, and heat, which it keeps us comfortable, but it can't be used biologically to do work. So light reaches the planet, and light is fixed. It is transformed. Light energy is transformed by autotrophs, principally by the plants, by organisms performing plant photosynthesis. And light energy is transformed into chemical energy, into the energy of bonds in molecules. The way in which that is done, there's something special about it, which I'll mention in a while. So by taking very simple compounds, carbon dioxide, water, using the energy from the sun and transforming it into high energy bonds, covalent bonds, plants generate organic molecules. And those molecules are what most of us as heterotrophs, this is what we use as our source, first of all, of molecules, but second of all, as our source of energy. And that we have to see that once these bonds have been made in these organic compounds, in order to access the energy in them, we're going to have to break those molecules. Likewise, when we take one set of molecules and we want to transform them into another useful set of molecules to do another type of work, we are going to have to put in energy. We're going to have to derive energy from somewhere in order to drive other processes somewhere else. And this is these are the continual processes of metabolism. That first of all, we have catabolic reactions. Catabolic reactions break down molecules, principally, not always, but principally to use them as a source of energy, to use the energy in their bonds as a source of biologically useful energy. And the second set of metabolic reactions are the anabolic reactions. The anabolic reactions require an input of energy. They are endogonic. Catabolic reactions are exergonic. Anabolic reactions are endogonic. They require an input of energy, which we derive from catabolism. So those are the driving cycles that maintain life on our planet. Today, we want to have a look at exactly how it is that we derive energy from these already created organic molecules. What is it about these molecules that makes them a source of energy? What does that energy look like in those molecules? And how do we access it? The way in which we access it is principally through a process called cellular respiration. And in eukaryotes, which is our principal interest, in eukaryotes, that process, the bulk of that process is performed in the organelles called the mitochondria. And in mitochondria, these organic molecules created originally by plants may be passaged through other organisms, maybe passaged through other things. Maybe we're eating a cow which once ate a plant. But nonetheless, what we are doing is we are accessing these organic molecules which have their origin in the plants. And we are now severing the bonds which they made in photosynthesis. And we are accessing the energy locked into them. And we do that through this process. It is called cellular respiration. And cellular respiration, we immediately think of, that is respiring, but it is not what we mean when we say respiration. Respiration is a very specific set of reactions which break down fuels, access the energy in the bonds of the fuels, and convert it into this substance here, ATP. We talked about it before. This is adenosine triphosphate. And most of the useful energy is locked into the bond of the last phosphate. This is adenosine triphosphate. Most of the energy that the cell will use ultimately is locked into the last phosphate bond. Can access some of the others, but usually it's just the last one. So there's the process. We all the 99% of the energy in the biosphere originates as light energy. The other 1% is there are some things which can use chemicals and other things as a source of energy, and still be autotrophs, but we're not going to worry about them. 99% of the energy which drives the biosphere is fixed by autotrophs using light. Light energy is converted into chemical energy, into chemical bonds. Those chemical bonds can be broken, and they're broken principally by this process of cellular respiration. And that produces a useful form of biological energy in the form of ATP. And the energy in ATP can then be used to fund endogonic reactions. Reactions like the anabolic reactions of building, where we need to put in energy. The point is We cannot simply break these molecules and directly use the energy to in endergonic reactions. We need an intermediate. We need a biologically useful intermediate to shuttle that energy, and that is ATP. ATP is the intermediate between catabolic reactions and anabolic reactions, between catabolic reactions and then the rest of the metabolism of the cell. I just point out, the process that we're going to be talking about, cellular respiration, it can be seen in all of the domains. We can see it in the prokaryotes. We see it throughout the eukaryotes, in the protists, everything else. It's a very, very, it's by far the most common way of producing ATP, but we will hear about other ways in which ATP can be produced. The process is essentially the same in all eukaryotes in the mitochondria. It occurs in the mitochondria no matter what eukaryote you're talking about. And we'll see how the structure of the mitochondrion has something to do with that. The mitochondrion is specially structured to perform this process of cellular respiration. But in the same process occurs in the prokaryotes, which of course have no mitochondria. And in the prokaryotes, the process is very, very similar. Don't forget, it evolved, cellular respiration evolved in prokaryotes, and we all in the eukaryotes have inherited it from the prokaryotes. In the prokaryotes, which have no mitochondrion, all the processes take place at the cell membrane, at the plasma membrane around the outside of the cell. Okay, so again, when we're talking here about accessing energy, which is built into these chemical bonds of the fuel molecules, and is stored there as potential energy in these strong covalent bonds. And in order to, what we're watching is how we're going to break those bonds and we're going to access the energy. But that begs a question, how do we access energy? What is the form of energy inside this cell? Inside those bonds, we say those bonds have energy. What do we actually mean? Do we break the bond and it flies out like a lightning bolt? No, it's not. It is in those bonds in the form of the electrons, of the energy inherent to the electrons, which constitute the covalent bond. That is where the energy is. And when we think about a fuel molecule in biological systems, just think about something like a carbohydrate. Think about glucose, C6H12O6, carbon with masses of associated hydrogens covalently bonded to it. How were those bonds made? They were made by changing sunlight energy into chemical energy. And the plants effectively do it by energizing the electrons of hydrogen. Those electrons of hydrogen get excited in the process of photosynthesis to the extent that they can actually form covalent bonds with carbon. And once they do that, that energetic excitement is locked into the activity of the electrons as potential energy. We can access that energy by accessing the electrons. And that is what we're going to watch. We're going to watch as actually electrons are transferred from food molecules to other molecules through a series of carefully controlled pathways. The pathways controlled all the way along by breaking them into steps, small steps, each governed by their own enzymes. When we talk about cellular respiration, remember all the way through what we are really talking about is dealing with electrons that we have derived from fuel molecules together with all of their energy, and then we're going to hand that on through steps in order to ultimately create biologically useful energy by taking that energy and put it into ATP. So when we talk about these systems, the first thing that we need to realize is we need a source of electrons, number one. We've got abundant electrons in things like the carbohydrates, also in lipids and things like that. We have abundant energy locked into all of those bonds between carbon and hydrogen. That's number one. We can access those by breaking those bonds, but then we need to transport those derived electrons. We need to transport them. So what we're going to need is we're going to need electron transporters. And the reason for that is that we're going to hand on these electrons. We're going to hand them on step by step through a series of reactions, which are referred to as redox reactions. When I take electrons and I add them to a substance, that substance we refer to as being reduced. The substance which donated the electrons, which lost its electrons, is oxidized. So those two reactions always coupled. We always have a substance which is being oxidized, losing its electrons, donating it to something else which is reduced. So we call them together, we call them the redox reactions. And if you want to think about it this way, whenever I increase the negative charge of a substance, I reduce it. Whenever I decrease its negative charge by removing electrons, I oxidize it. And the two are always coupled. What we're going to watch is how electrons derived from the hydrogens in fuels are handed on through series of redox reactions in order to ultimately harvest the energy. The reason for that is that at the end of our systems of cellular respiration, which we're going to talk about today, there is one master molecule which is sitting at the end saying, "Come to daddy," and that is oxygen. Oxygen is extremely electron negative, extremely attractive to electrons. And what we're going to see is that we're going to take fuel, we're going to strip off hydrogens together with their electrons, we're going to carry those electrons into a series of reactions, of redox reactions. And as they pass through all of the redox reactions, every time they go through a redox reaction, they will lose some of their energy. They will pass from a high energy state in the fuel to a much lower energy state at the very end. And at the end, they become joined to oxygen, which is pulling them through these reactions. Those electrons will ultimately end up joining to oxygen. They don't just join as electrons, they will join together with the nucleus of the hydrogen atom, which is a proton. And we join hydrogen atoms to oxygen, we end up producing water. So water is an end product. But the ultimate electron acceptor in this whole system of cellular respiration is oxygen. That's why you're breathing oxygen right now. You're breathing oxygen to drive all of your processes of cellular respiration. Okay, so let's just think about the actual chemical background to the processes. So cellular respiration, you'll also very frequently see the term interchanged with aerobic respiration. Aerobic respiration, because as I told you, oxygen is sitting there at the end of the whole process as the ultimate electron acceptor. We're stripping electrons at the start, sending them through a whole series of reactions, but they have to have a final destination, and that final destination is oxygen in aerobic respiration. So the cell can use many things as a source of fuel. You can use carbohydrates very commonly. Don't forget, in plants, the principal storage product is starch, which is a carbohydrate. In animals, the principal storage product for immediate access is glycogen, which is also a carbohydrate. But we can also use, our long-term storage fuels are things like lipids and oils. In some circumstances, we can use proteins and various other things as fuel as well. But the easiest way to follow what is going to happen is to use the simple molecule of glucose, which is perfectly appropriate because glucose is very frequently used as a fuel in the cell. So this is glucose here, C6H12O6. C6H12O6, look at that. We've got 6 carbons. We've got all of these hydrogens and 6 oxygens. This is what we're going to be really interested in, because a hydrogen atom is very simple. It just consists of a nucleus which has 1 proton, and it has 1 electron, which is quite loosely associated with that proton. And that electron is very easily excited. It can be pushed to higher energy levels very simply. And when plants make this substance, they boost the energy levels of hydrogen atoms to form this substance, to join the hydrogen in water to the carbon in carbon dioxide to make the substance C6H12O6. These bonds here contain a lot of energy locked into high energy level electrons. So this is what we effectively do in respiration. We take glucose, we take oxygen, and we, say in inverted commas, burn it. And we end up with its original components, which is carbon dioxide and water. This is a balanced equation. We end up with carbon dioxide and we end up with water. But in addition, we end up with energy. Now, let me tell you something. If I was to take a bowl of glucose, of crystalline glucose, I could actually set a light to it on a bench. It takes a little bit of going. It actually requires quite a high energy of activation if you want to burn it. But once you get it going, it will burn like any fuel. And if we were to watch it, it would just burn uselessly. It would produce light. We could see it burning by the flame, and it would produce a lot of heat as well. Neither of those are biologically useful forms of energy. But still, I would end up being able to produce this balanced equation if I just burned it with a flame. I could take glucose, burn it with oxygen, and end up with carbon dioxide and water and useless energy, biologically useless energy, light and heat. This metabolism of the cell does effectively the same thing. It effectively burns glucose with oxygen, but instead of doing it in a useless, chaotic fashion like that, it does it in a carefully controlled, stepwise fashion, carefully shuttling these electrons so that they perform useful work and end up producing ATP. The cell can't do it completely efficiently. It only does it with about 30% efficiency. But so it has to, unfortunately, we produce a lot of waste heat as well. But nonetheless, we derive most of our biologically useful energy from exactly the same process. The reason that this has to be done like this is, as I told you a minute ago, the catabolic reactions, burning fuel, like burning glucose like this, we can't burn it and just directly access this energy. We can't use light or heat. Instead, it has to be, some of this energy has to be transformed into a biologically useful form, and that is ATP, in general, ATP. Sometimes in the cell you will encounter other, like GTP, but that's not very common. It is usually 99% of the time, it is biologically useful energy comes in the form of ATP. The ATP is a very unstable molecule. it's not long-lived at all. So the cell makes ATP immediately it needs it. As it needs it, it makes the ATP, and that funds the reactions of the anabolic pathways, is used immediately. This means that we need constant supply. We continually need to regenerate ATP. If we use up ATP in a biologically useful reaction, we end up with ADP and free phosphates usually when everything is over and done with. So we continually produce ADP from ATP and harvest the energy. So we need to continually regenerate ATP. And we do that by performing this process. This is cellular respiration or aerobic respiration, aerobic because it's using oxygen. And those are the processes that we're going to follow today. So we're going to see that what we're effectively doing, look, we've got a fuel molecule with lots of hydrogens. If we're taking off the hydrogens and their electrons, we're removing electrons from it. We are oxidizing the fuel molecule. And fortunately, our fuel molecules, things like the sugars, even lipids, those sorts of things, have abundant hydrogens covalently bonded to a carbon, so they're excellent source of electrons together with their attendant energy. And we are going to see that is exactly, we are performing a system of redox reactions. And ultimately, when we look at the whole process together, we can think of it in terms of a redox reaction, remembering that there are a whole lot of intermediate reactions, okay, in between. But just think about it this way. We've got a fuel, in this case, glucose, which we oxidize. There, look there. Why have we say we've oxidized? We've oxidized it because we've removed the hydrogens. We've moved, not only removed, we have moved the hydrogens with their attendant electrons. It's the electrons we're really interested in. We have oxidized a fuel source. And we have reduced, ultimately, at the end of the whole process, we have reduced oxygen. Why have we reduced oxygen? Because we've added the electrons to oxygen in a covalent bond. We have reduced oxygen. In that process, in that redox reaction, energy has been released. The electrons here have moved from a very high energy state to a low energy state here. And that energy is released. And in biological systems, it is released stepwise, and the energy is harvested in order to produce ATP. So just think about this here. I said we've removed these electrons, right? We end up putting them here. We end up reducing the oxygen. We've removed electrons. Well, the electrons don't fly across in a lightning bolt. Instead, we need to be able to shuttle them. We need something which will actually carry electrons for us from one reaction to another. And that we refer to those electrons with their attendant energy as being reducing power. So we need a source of reducing power in the cell. We need some way of taking the electrons associated with hydrogen and actually shuttling them through the cell to the appropriate reactions in order to harvest their energy. The shuttle we use is typically this one here. This is called, it's a complex compound. You don't need to remember the structure. Just remember the name. It is nicotinamide adenine dinucleotide. And if you look at here, just look at this part here, and you'll realize that looks like a nucleotide. It is. But NAD is a dinucleotide. It's two nucleotides joined together here. It's called nicotinamide adenine dinucleotide. And it is in its native form, it is positively charged. So what this makes it very suitable as an electron shuttle, because it can easily pick up 2 hydrogens. So let's just do it. It's plus, right? So it's lacking an electron. It's lacking 1 electron. So I'm going to take 1 electron from, first of all, let me just specify. I'm going to use a hydrogen molecule, not just an atom, a molecule, H2. And that means it's got 2 protons, 2 electrons associated with it. I'm going to take 1 of those electrons and add it to the NAD. Okay, that's going to mean it becomes just NAD. It's now uncharged. I've added an electron, and I'm going to bond a hydrogen to it. I'm going to a whole hydrogen atom, proton and its electron. What have I done? I've effectively added 2 electrons and a hydrogen atom to it, and I end up with NADH. NADH is the reduced form of NAD+. Just remember that, NAD plus hydrogen atom, H2, goes to NADH, and we have associated with that a spare proton, H+. That is the remainder. We stripped its electron, put it onto the NAD+, and we added a hydrogen, but we left NADH plus a proton. And NADH is one of the most important electron shuttles in the cell. We're going to see NADH used over and over again. I should tell you, it's not only used in cellular respiration, it's used in other things as well. But we're going to watch it in particular as it's used in cellular respiration. So just this complicated diagram, what I want you to remember is NAD plus is the oxidized form of nicotinamide adenine dinucleotide. When we reduce it, we actually add 2 hydrogens, and we end up with the reduced form, NADH, and a proton there. You're going to find out that these protons are very, 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 very useful. They are produced all the way along the line, and they are extremely useful in the process. So just to emphasize, for this entire reaction, and taking a fuel, combining it with oxygen to produce energy and terminal products, carbon dioxide and water, we could do it catastrophically. As I told you, we can light glucose. We didn't talk much about free energy changes, but it has a high free energy change. It becomes extremely, the change in free energy is strongly negative, which means it's a strongly exergonic reaction, produces a lot of free energy. If I simply do it, light it, it is catastrophic. It releases heat and light energy, but there is a free energy change. When we look at how it's done in the cell, we see exactly the same free energy change. We can harvest all of this energy, and that is done by passing it through a series of redox reactions. What we're going to see is that we're going to shuttle our electrons in the form of NADH and another electron carrier that we'll encounter in a minute. We're going to shuttle our electrons to a system of controlled redox reactions. They work like this. We're going to pump in electrons here and reduce this substance. That substance in turn is going to be oxidized and donate its electrons to the next. This is reduced, and then it becomes oxidized, hands its electrons onto this one. This one is reduced, hands its electrons onto this one. And ultimately, the electrons are handed off to oxygen. And in that step, as we pass through these steps, each time we go through one of these steps, the electrons drop in energy. And the energy is not just lost. The energy is actually harvested. And the energy is harvested to make ATP in a very cunning way that we'll encounter in a minute. Okay, so the whole process, we're going to follow a glucose molecule, one glucose molecule. And the whole process, it goes through a series of complex steps. We are not going to consider all of the detail, nor are you going to be asked to remember. I will specify exactly what I need you to remember in each of the steps. But, excuse me, just remember that the reason is broken down into these very complex metabolic pathways is precisely so that the breakdown of our fuel is done in a biologically useful way, that it is not a catastrophic release of an explosive release of energy. Instead, we're seeing a stepwise harvesting of energy from the electrons of the fuel. And what we'll see is that there are two principal locations for these reactions. The first occurs inside the cytoplasm, and that is a process called glycolysis. Glycolysis literally means breaking glucose, and that's exactly what it does. The next step, from then on, everything takes place inside the mitochondrion in eukaryotes. In prokaryotes, all of these processes take place at the plasma membrane, the cell membrane around the outside of the cell. In eukaryotes, the products of glycolysis are imported into the mitochondrion. And we then go through, there's a little intermediate step, and then they pass into a major cycle here. And as we see the products of these various reactions, what we notice, first of all, at this step here and at this step here, glycolysis and this step here, we make a little bit of ATP through a process called substrate level phosphorylation. That simply means that the ATP is enzymatically made by taking a phosphate, taking an ADP molecule, and putting them together enzymatically as a simple chemical reaction. That distinguishes substrate level phosphorylation, is distinguished from oxidative phosphorylation, which is the process that occurs at the end. And you'll see why. Oxidative phosphorylation is quite different to substrate level phosphorylation. Substrate level phosphorylation is much more like just a simple chemical reaction, ADP plus P, ATP, done by an enzyme. This is a much more complex process, as we'll see. What is important about all of these reactions? First of all, yes, these reactions, glycolysis and the citric acid cycle, do produce a little bit of ATP, but they don't produce very much. Not very much at all. Okay. But what they do produce is masses of NADH. Look here, NADH there, and then masses of NADH here at the citric acid cycle. And that's what's important, because it's the electrons carried via NADH and this other electron carrier I'll tell you about now. Those electrons are going to be shunted here to a very, very, very efficient means of making ATP. In this process here, all of the energy, a lot of the energy associated with those electrons is going to be harvested to produce ATP. And what we're going to end up with at the end of all of these processes, all of them, we're going to end up with up to 32 molecules. In bacteria, sometimes even higher, can go up as high as 38. In eukaryotes, it's usually around about 32. There's some variability. But remember this number, 32. Just get that in your head as the standard. For one molecule of glucose, we produce 32 molecules of ATP. That's a good average to remember. Okay, so we've got one part in the cytoplasm, the rest inside the mitochondrion. And let's just talk about this very first process of glycolysis. And glycolysis literally means breaking glucose. Glycolysis is a process which evolved in the prokaryotes, and it evolved at a time when there was no oxygen on the planet. It's the original, it's a holdover of the original processes that the cell evolved to produce ATP. And again, it's a complex process. There are many, many steps to it, but I'm not calling on you to remember all of the steps or all of the intermediates. I'll make clear to you exactly what I want you to remember. Here's the whole process if you're really interested. Here it is here. If you are inclined to studying biochemistry, then you can do so, study the intermediates and all of their enzymes. I simply want to point out here, we're going to start off with glucose. And what we're going to do is we're going to actually phosphorylate the glucose there. You can see it's glucose. Now it's been phosphorylated. It turns into a completely different substance there. But this substance here is very unstable compared to the glucose itself. It's very unstable, and it's easily broken by enzymes into two separate molecules. We're going to start off with a 6-carbon glucose. We're going to produce 2 3-carbon, 1, 2, 3. We're going to produce 2 3-carbon compounds. Don't worry about this step here. Just remember this glyceraldehyde 3-phosphate, G3P. We're going to produce 2 molecules of G3P. Okay, and here's our glucose. We want to actually destabilize this glucose. So what we're going to do is we're going to invest 2 ATPs, ATP to ADP. What happens to this phosphate is bound to the glucose. Very high energy, by the way. And it starts to destabilize this. It energizes this molecule. We're going to invest another one. Here it is here, ATP to ADP. Now we've got our double phosphorylated molecule there. And that molecule is then broken into these 2 molecules, glyceraldehyde 3-phosphate. This we call the energy investment stage. Here it is here, glucose, 6-carbon compound, right? We're going to add 2 ATPs in order to double phosphorylate that. And we used up 2 ATPs. It doesn't sound like a good idea, but what we do is we've actually made 2 molecules of G3P. We've added 2 molecules of G3P, and we're going to follow both of those now. And here, in this series of reactions, we are actually going to be able to harvest some energy, and we're going to harvest it in 2 forms. So look here, we produce 2 molecules of glyceraldehyde 3-phosphate there, right? We're going to follow each. Remember, this is going to now run twice because we've got 2 of these molecules. This is following one of those molecules, and here it goes. Look at the very first step, and what do? We make NADH. They've doubled this up because in actual fact, they're following 2 molecules of G3P. In the reactions, we're going to make 2 molecules of NADH. What is NADH? It's carrying high energy electrons. There's our energy that we're interested in. This step here, we actually make 2 ATPs, right? And that balances these 2 that we had to invest. This step here, we make another 2 ATPs. So we gain 2 ATPs. This whole pathway, what we've done is we've taken glucose, destabilized it by phosphorylating it, broken it, and we've ended up being able to harvest hydrogens together with their energetic electrons. And we've been able to make 2 molecules net of ATP. This process, glycolysis, for some organisms is enough. This is enough of a source of ATP for them to actually fund all of the processes of the cell, usually at a low metabolic rate, but nonetheless, some organisms, as we'll hear, can manage just with glycolysis. And those are the anaerobic organisms that do not utilize oxygen at all. They can depend on glycolysis as their only source of ATP. But for all the rest, this product here is what we are really interested in. And that is a substance now called pyruvate. And don't forget, from one molecule of glucose in glycolysis, we end up producing 2 molecules of pyruvate or pyruvic acid. And we're now going to follow pyruvate and see what it, and look, how many carbons does it have? 1, 2, 3. We started off with 6 carbon atoms in glucose. Here are our 6 carbon atoms, 1, 2, 3, but we have 2 molecules of pyruvate, 6. In the eukaryotes, this pyruvate is then imported into the mitochondrion. It is actively transported into the interior of the mitochondrion. Remember the structure of the mitochondrion. The mitochondrion has an outer membrane around it. It is double membrane. It has an outer membrane and it has an inner membrane folded like this. So it has its own, it has equivalent of its own cytoplasm in here. And this membrane here has an inside and an outside. And we're going to see that that's very, very important because this membrane acts as a boundary. And all of the enzymes that we're going to talk about in this process here are on this membrane here. The processes I'm going to describe now take place inside the equivalent of the cytoplasm of the mitochondrion in here. The first, remember, what are we doing? We're importing this 3-carbon compound called pyruvate. We import it into the mitochondrion, into the inside of the mitochondrion, and it undergoes an intermediate reaction. And it's often called the intermediate step. Here's the pyruvate coming in, and the pyruvate is dehydrogenated. Look at the name of this enzyme, pyruvate dehydrogenase. As soon as you see dehydrogenase, you know that what is happening is hydrogen and its attendant electrons are being shuttled. They're being moved from one substance to another. And the substance they are being moved to here is NADH. We had some NADH produced in glycolysis. Here we're having more NADH produced. And remember, always there's an attendant proton. If we reduce NAD+, we end up with NADH and an attendant proton, a resulting proton. So pyruvate dehydrogenase also breaks pyruvate into a 2-carbon compound. And 1 carbon there is released as carbon dioxide. Breathe out. What are you breathing out? You're breathing out carbon dioxide. Some of that carbon dioxide originated here. It originates in your mitochondria. The first lot of carbon dioxide originates from this intermediate step. And the intermediate step breaks pyruvate and binds it to this substance called coenzyme A. You just need to remember this name here, acetyl coenzyme A. So remember the products of the intermediate step. The products of the intermediate step are NADH and a proton, carbon dioxide, and acetyl coenzyme A. And remember, for each glucose molecule, 2 pyruvates come in to go through this intermediate step. The acetyl coenzyme A is important because it's the only way that we can get these carbons and the attendant hydrogens into the next series of reactions. And the next series of reactions are actually a cycle. It's called the Krebs cycle, or named for the person who first described it. But more properly, it's called the citric acid cycle, because citric acid is the very first product of the cycle. So the citric acid cycle, again, takes place within the mitochondrion, and we're going to see it produces some ATP, not a lot, but it produces some ATP, but it produces masses of reducing power in the form of the electron carriers. So here is the complex cycle. Again, I do not expect you to remember everything, but I want you to look carefully at what happens at the process that we're watching. Remember, we said that intermediate step produces this 2-carbon compound called acetyl coenzyme A. That enters the cycle here, and it binds with this substance here called oxaloacetate. And oxaloacetate is a 4-carbon compound, 1, 2, 3, 4. And so we end up with this 6-carbon compound called citrate. So this is what I want you to remember. Acetyl coenzyme A enters, binds with oxaloacetate, and forms citrate. It is a cycle because all of these steps in this metabolic pathway end up regenerating oxaloacetate at the end. Okay, so we've got acetyl coenzyme A joins oxaloacetate, forms citrate, goes through the cycle which regenerates oxaloacetate. But it's what happens in the intermediate steps that really interests us. Have a look here. Here's a place where we harvest hydrogens, NADH. We are going to reduce NAD plus again, NADH plus a proton. Here again, NAD plus goes NADH plus a proton. This here is another electron carrier called flavine adenine dinucleotide. And FAD, this is reduced to FADH2. So there you can see it's carrying an entire hydrogen atom from FAD to FADH2. It functions much the same as NADH. It's an electron shuttle. So we produce FADH2, and here we produce more NADH. How many NADHs did we produce? 1, 2, 3, plus 1 FADH2. We've produced 4 electron carriers, and we're going to do this twice. Why? Because we broke glucose into 2 pyruvates. Pyruvates went to 2 acetyl coenzyme A's. So for every glucose molecule, we run this twice. In addition, we produce a little bit of ATP here. In addition, if we go through, you'll find that this is also the source of much of our carbon dioxide. What we're doing is we're taking whatever we're feeding through here, and we're actually oxidizing it. Okay, we're oxidizing it by passing hydrogen and electrons onto NAD plus here, for example. We've stripped hydrogens off of carbon, and we expel that carbon as carbon dioxide there and carbon dioxide there. So our sources of carbon dioxide are the intermediate step and now the citric acid cycle. So that's all the carbon dioxide that you're breathing out, okay, is derived from those processes. Remember the products NADH and protons and FADH2 and a little bit of ATP. So what we're really interested in is this. All of this reducing power, all of these electrons derived now originally from our glucose fuel, but now put into these electron carriers and humming with energy, humming with energy originally derived from the sun's energy. And now we need to try and harvest that energy. And we do it in, we're going to do it in this process called oxidative phosphorylation. This gives you, by the way, a summary of all of the products. So this is what I want you to really remember. Okay, this is the products after glycolysis. So after glycolysis, we 6 molecules of carbon dioxide, 12 molecules of NADH, 12 protons, 2 molecules of FADH2, and 2 molecules of ATP. Okay, add that to the 2 ATPs and the NADH that we produce in glycolysis as well. So those electron carriers, NADH and then FADH2, those are what we're really interested in now. We're interested in the hydrogens that they are carrying with them, because those hydrogens have attendant electrons at a high energy level. And we're going to now pass them through a series of reactions where that energy level drops, and we're going to harvest the energy. And we're going to harvest it in a very interesting way. And so this is a complex process. The electrons actually pass through a series of substances which will pick up the electrons in a pairwise fashion. They pick them up and are reduced, and instantaneously they're going to hand off those electrons to a neighboring substance. So they are going to be oxidized and reduce the next substance. And this goes through a series of complex enzymes and carriers and things. We're not going to worry about all of the details of the names and everything in the pathway. I'm just going to show you these depend on proteins which are integral to the membrane. So these proteins in a eukaryote, these proteins are integral to the inner membrane of the mitochondrion. And it's important to keep note of what is on one side of the membrane and what is on the other. And to remember that the membrane is a very profound barrier. It's a very good insulator, because all sorts of things are going to happen, because we're going to start accumulating stuff on one side that isn't on the other. There's a tremendous amount of energy bound up in NADH and FADH2. And this process is quite, in biological terms, quite efficient. In overall terms, not that efficient, only harvests about 35% of the energy as useful biological energy. But nonetheless, it is far more efficient at making ATP than any of the other processes that we've talked about so far. So we go back, please, and have a look at the mitochondrion and look at its structure again to remind yourselves about the relevance of the inner membrane related to the outer membrane and the spaces that arise because there is an inner membrane. So here is just a simplified diagram, and you see it's a complex set of proteins which are embedded in the membrane or associated with the membrane. Peripheral here, but intrinsic there. And many of them are intrinsic. I just want to point out several things to you. First of all, what we're going to do is we're going to actually donate the electrons from NADH, from the hydrogens in NADH. We're going to donate those electrons to this protein. That's our first one here. These proteins can have multiple roles. In this case, this first proton has 2 roles. First, it is an electron carrier. It will temporarily be reduced, and it will then hand off those electrons to another substance here. This substance, in turn, is reduced and is then oxidized to hand off the electrons here. But this substance here not only transports those electrons effectively, it is also a pump. And the passage of the electrons here, that oxidation reduction reaction, the electrons drop in energy level, and that energy is harvested by this substance, which is, and the energy is used to pump protons. There they go there. It will pump protons. As those electrons pass through, it will pump the protons to the other side. This here is the interior of the mitochondria, the innermost part of the mitochondria. This is the space in between that outer membrane, and this is the inner membrane. So what is going to happen? We're going to pump protons. Well, when you pump protons, all sorts of things result. If I add protons to an aqueous solution, what am I doing? I'm lowering the pH. I'm making this acid compared to this. If I pump protons, what am I doing? I'm pumping a chemical H plus hydrogen to this side so that there is more of it on this side than there is on that side. Number two. Number three, if I pump protons, I'm pumping charge. I'm creating a charge differential. There is much more positive charge on this side than there is on that side. What I've done is I'm creating a very profound gradient. There's a chemiosmotic gradient. It's also a gradient of charge. There is a charge differential here that wants to equalize. There is a chemical difference there and a pH difference which want to equalize by diffusion. So by pumping protons across, I create a tremendous amount of potential energy. I created a chemiosmotic gradient which inherently needs to be satisfied somehow. Okay? It creates a potential energy across this barrier. There are, in fact, 3 of these proton pumps, there, there, and there. And the electrons, as they flow through this chain of substances, mostly proteins, not all, but mostly proteins, as the electrons flow through here, they fall in energy as they approach oxygen at the very end, is harvested to pump protons. And that creates a tremendous amount of potential energy across that membrane. So that is NADH puts its electrons in here, into the first one. That other carrier that we talked about, FADH2, it also transports its electrons and it pumps in a pair of electrons, but it's a little bit further down the chain. It's not, and so it does. The passage of these electrons doesn't result in the pumping of as many protons across the membrane, but nonetheless, very rapidly, this gradient is set up, and that potential energy can be harvested. And we're going to harvest it to actually manufacture ATP. And the way in which we do it is we use this. We use that chemiosmotic gradient and allow the gradient to equalize through this very, very complex protein, which is called ATP synthase. And that should tell you immediately. ATP synthase, it's an enzyme. What is it going to do? It's going to manufacture ATP. It does it in a very peculiar, complicated way. But let's just remind ourselves of where we are. From all of our other processes, glycolysis, the intermediate step, the Krebs citric acid cycle, we created a little bit of ATP by substrate phosphorylation. But the most important thing was we created this NADH and FADH2. Why are they so important? Because they are shuttling electrons, high energy electrons. and they are donating those electrons. They themselves are being oxidized. NADH is oxidized to NAD+. FADH2 is oxidized to FAD. The electrons that they carried were donated to this. The protons that resulted from these reactions are pumped across. Remember, NADH has associated with a proton. And same thing if I oxidize FADH2, if I strip off the electrons, I'm creating protons on this side. Those protons are pumped across this membrane. They are pumped here, here, and here, which rapidly builds up a gradient of charge and a chemical gradient. That is a chemiosmotic gradient. That chemiosmotic gradient now is going to be used. What is the fate of the electrons that are passing through here? They are attracted at the end to oxygen. And what we're going to find is that they are going to unite with oxygen and protons to form water. But we still got to equalize the proton balance. We've got to get these protons back across here. And we do it by allowing them to pass through this complex enzyme here, ATP synthase. This is the way it works, just in very, very briefly. Here's the intermembrane space, okay? That is between the outer membrane and the inner membrane of the mitochondria. It is full of hydrogen ions. It's very acid. And it's got lots of hydrogen ions and very few on this side. The ATP synthase has a channel right through the middle. And the protons can actually flow through the ATP synthase in a complex way. They bind to it and then pass through it and are pumped to this side here. So they are equalizing that gradient by a chemiosmotic process. As they move down, the energy, the potential energy that they contain is harvested by the ATP synthase to synthesize ATP. It takes ADP, phosphates, and it produces ATP. And this gradient is satisfied. In addition, when it does that, the protons flow back into this mitochondrial matrix, and they, together with their electrons and with oxygen, are united to form the ultimate final end product, water. Oxygen is the terminal electron acceptor. We had an intermediate electron acceptor in the carriers. We had intermediate electron acceptors in this chain, but we have an ultimate electron acceptor in oxygen, and that forms water. That's why you breathe out water. You breathe out carbon dioxide from all those previous reactions. And that is the end of the process. And we end up producing very, very efficiently. Compared to our earlier reactions, we end up producing a lot of ATP for each glucose molecule. So here is the sum total for you. And glycolysis, we put in 2 and we got 4. So we had a net of 2 ATPs. The citric acid cycle, we got another 2 molecules of ATP, not very much. But these processes here produce a whole lot of electron carriers carrying energized electrons, which went to oxidative phosphorylation. And that process alone, oxidative phosphorylation, produces between 26 and 28 molecules of ATP. You can accept, just stick with 28, it's fine. There's a little bit of variation in how much. There's some variability in the structure of the chain. Some chains produce more, some chains produce less. There's some variability because actually the protons can be taken off and used in other processes. And there's various other things which can influence this. the efficiency of the oxidative phosphorylation. But for our purposes, just accept 2, 4, 28, 32 molecules of ATP altogether from one molecule of glucose. Okay, so I think I'm going to leave it there for the moment. And I'll, next class, I'll just discuss the last part of this, which is fermentation. And that will be it. We'll then also talk briefly, very briefly, unfortunately, but briefly about this fantastic process of photosynthesis to which we owe all our lives. Okay, I hope that this is comprehensible to you. Go slowly through what I have done. Stop the video every 10 minutes, 15 minutes. Check your notes. Go back over what you don't understand, and then proceed. Do it slowly and repeat it. Repeat often. The more you repeat, the more you'll understand, the more you'll comprehend, and the more you'll remember. Okay.